Hello, this is Sudarshan, and in this episode, we'll talk about chapter 41, that is Enterobacteriaceae family that causes gastrointestinal infections. So, first coming to the characteristics of Enterobacteriaceae, as the name itself suggests, these are the group of bacteria which will be causing infection in the intestine, and the properties are very important. We have gram-negative bacilli, facultative anaerobe, they are non-fastidious, that is they can grow in the cultures, and they are glucose fermenters, then they are catalase positive and oxidase negative. They have the peritrichous flagella that is surrounding the organism, flagella is there, right. And with respect to, okay, so this is with the characters. Now let us see with the infections. So first starting with E. coli. So E. coli, it is a normal commensal in the intestine, but sometimes when the infective dose is more, it can cause infections. So first we'll see the surface antigens. So for the surface antigens, let us have a mnemonic HAPCO. First H for H antigen, that is responsible for the motility or the movement of the movement of the E. coli, and A is nothing but lipid A that causes endotoxic shock, and P is nothing but the P. pili, which is present in the lining epithelium of the urinary tract, that mainly causes cystitis and pyelonephritis. And then K, that is the capsular antigen, mainly targeting the lipopolysaccharide capsule. Mainly this is responsible for meningitis and also pneumonia. And O, that is the somatic antigen, mainly related to the antibodies formation. And okay, so this is with the different antigens. Now let us see with the toxins. So first of all, we have this heat labile, heat stable, and Shigella toxin. Let us see. So heat labile, as the name itself tells, it is resistant to the heat. Heat labile means it is destroyed by heat. Heat stable means it is resistant to the heat. And Shigella toxin. So E. coli makes a toxin very similar to Shigella. So we have here, right, so we have here LT and ST, that is the heat labile and stable, produced by ETEC, that is Enterotoxigenic E. coli. As the name itself tells, this is a E. coli that produces a harmful substance, that is a toxin that harms the intestinal cells. Now let us see the details. So coming to heat labile and Shigella toxin, they look very similar. Both will have two fragments. So let us see the differences. In the first fragment, in case of heat labile, it will basically cause ribosylation in the ADP that activates the G protein. Whereas in case of Shigella toxin, A will inhibit the 60S ribosome. And with respect to the B fragment, in heat labile it binds to GM1 receptors. In the heat stable, in the Shigella toxin, it binds to GB3 or globotriazyl ceramide receptor on the epithelium. And with the heat stable, it binds to cyclic GMP that causes diarrhea. So both heat labile and stable are plasmid coded, whereas Shigella toxin is bacteriophage coded. Right, and all these three toxins can be detected by ELISA PCR. Now coming to the important toxins associated with E. coli, very important. First is EPEC, EPEC or Enteropathogenic E. coli. Second is ETEC or Enterotoxigenic E. coli. Third is EIEC or Enteroinvasive E. coli, and then EHEC or Enterohemorrhagic E. coli. Then we have EAEC or Entero-Aggregative E. coli, and the last one, that is DAEC or diffusely adherent E. coli. So let us see with one by one the strains. So firstly we have EPEC or Enteropathogenic E. coli. As the name itself tells, this strain of E. coli causes disease in the intestine. Right. And next we have the Enterotoxigenic E. coli. As the name itself tells, this form of E. coli will produce a harmful substance called a toxin that will harm the intestinal cells. And very important, Enterotoxigenic E. coli or ETEC causes traveler's diarrhea. Then EIEC or Entero-invasive E. coli. As the name tells, this is a form of E. coli that invades the entire intestine and it will damage the intestinal cells. And there is something called a VMA that is important, virulence marker antigen. Then EHEC or Enterohemorrhagic E. coli. As the name itself tells, this strain of E. coli causes bleeding in the intestine. Right, and it secretes the Shigella toxin. So Shigella toxin is important with EHEC. Right, and here importantly it causes HUS, that is hemolytic uremic syndrome, which has a triad of features, that is MAHA or microangiopathic hemolytic anemia, thrombocytopenia and renal failure. And then EAEC, Entero-Aggregative E. coli. So as the name suggests, this is a form of E. coli where all the E. coli will come together in the intestine and bind to the cells, produce the toxin and damage it. So actin-based motility also can be seen here. Right, and finally we have DAEC or diffusely adherent E. coli. As the name itself tells, the E. coli will attach to one intestinal cell and then spread the disease to the other intestinal cells also. And when it comes to the diagnosis, mainly we will do the toxin detection, the molecular methods, the automated techniques like MALDI-TOF-Vitek, and then we will do a culture to detect the microbe. We'll do a sample collection based on the site affected, since sometimes disseminated infection also is there. Then we will do the biochemical tests also, and treatment would be supportive care. And next, coming to Shigella, the next member of this family. So there are 4 important species of Shigella, that is Shigella dysenteriae, then S. flexneri, S. boydii and S. sonnei, SONNEI. So S. dysenteriae is important. As the name itself tells, this will cause a disordered intestine. It will cause an intestinal disease. So let us see with the pathogenesis. So Shigella will try to enter into the M cells of the intestine that is present in the Peyer's patches, which is having the malt mucosa associated lymphoid tissue. So the macrophages will be there as soon as it detects the presence of the Shigella in the M cells. It will try to engulf this bacilli, but it will develop resistance, Shigella. And so through the apoptosis, all the Shigella will be bursting out and cause a disseminated infection. And obviously it secretes an enterotoxin, and the actin-based motility will be present here, and it doesn't ferment lactose and it doesn't form H2S. This is important. Right, and the most important species of this class is S. flexneri, that is the most accounting into number of cases. And with clinical features, we are going to have GI related symptoms and a flu-like illness, that is fever, malaise, myalgia, arthralgias, etc. And coming to the diagnosis, molecular techniques, ELISA PCR, automated techniques, MALDI-TOF-Vitek, culture, then sample collection, mainly stool sample collection will be done. And with respect to the treatment of Shigella, it will be supportive care. And this Shigella is very common in daycare centers. So there must be good food hygiene habits. And next, coming to Edwardsiella, the most important species E. tarda or Edwardsiella tarda. Same diagnosis and treatment methods. And then we have the salmonellosis caused by salmonella. And we have two important species of salmonella. One is Salmonella Enterica, another is Salmonella typhi. So here we'll be talking about Salmonella Enterica only. As the name suggests, this species of Salmonella will affect the intestine. And it has both animal and human reservoir, and mode of transmission is mainly through the fecal-oral route. And with respect to the clinical features, it causes gastroenteritis, that is the infection and inflammation of the stomach and intestine, and also a disseminated infection as well. And with the diagnosis, we have the sample collection, that is the stool sample, molecular techniques, automated techniques, antimicrobial tests can be done. And with respect to the treatment, for Salmonella Enterica, there is no antibiotics and vaccination generally given because of drug resistant strains. So supportive care can be done. And then we have the Yersinia infections. Mainly two species are there. One is Yersinia Enterocolitica, another is called Yersinia pestis. The pestis one will cause bubonic plague. Here we will see why Enterocolitica. So as the name suggests, it will affect the colon and the intestine. And with the pathogenesis, obviously it has to infect the intestinal cells, and mainly it uses two proteins here, or two molecules called YADA, Y-A-D-A, another is AIL, and also it uses something called a siderophore, where basically it reduces the iron deposits in the body. So it causes iron deficiency anemia mainly. Right. And this Yersinia Enterocolitica mainly causes pseudoappendicitis, that is the false inflammation or the infection of the appendix, and very commonly it will infect the mesenteric area of the intestine, where we have the fat, the lymph nodes, the neurovascular structures. So mainly the mesenteric area of the abdomen is affected. Right. And also it causes the HLA-B27 associations, very commonly reactive arthritis. The same with Bartonella species also. And with the diagnosis, culture can be done on McConkey agar, where we will see the pale colonies because it doesn't ferment lactose. The same with Shigella, where we see pink colonies on McConkey agar. And apart from that, automated techniques, serology, etc. And treatment, mostly supportive care. Antibiotics are indicated for severe infection. And there is another species in this class, that is Plesiomonas. So that is all with the Enterobacteriaceae family. So thank you so much. We'll see you in the next video.